Global temperatures have risen by 1.5 degrees Celsius. Sounds small, right? But if your fridge did the same, your yogurt would turn into a biology experiment. And guess what? Our urban trees are struggling just as much. Scientists have a wild plan to save them. Let me explain. Fun fact, plants don't move, or at least not like us. So to cool themselves down and fight against drought, no AC or a dive in the pool, but other strategies that are very different. First, absorbing more water makes sense, right? A second one, avoiding the loss of water. Or even a third one, producing compounds that fight against the molecular damage that happens during a stress of that type. You know, when you struggle, you can give a call to a friend. For the plant, it's the same, and its friends are microorganisms. The soil is full of them, from bacteria to fungi. In our case, we will focus on fungi, and more precisely, one group, the endophytes. In some of them, we can find compounds similar to the ones the plant produces in a drought period. And this could really help the plants. Want to know more about our project? Grab some popcorn and let us guide you through our scientific adventure. We will work on oak species that are resistant to drought and others that are not, from all over the world, and try to multiply them. We will sterilize and put to germinate embryos of oaks under in vitro conditions. We expect them to grow shoots. This process will be followed by a multiplication phase using cuttings of these shoots in order to regenerate apexes. After that, we will transfer them in a medium which encourages root development. We will make them grow and finally acclimate the trees to ex vitro conditions. And that's, my friend, how you propagate trees. In the end, from one seed, we could produce dozens or even hundreds of trees. This is what we hope to produce. Since endophytic fungi can produce compounds that can help the plant to resist drought stress, we will study two strains that will help the plant in two different ways. Periconia macrospinosa should enhance the resistance to oxidative stress, which is increased when plants suffer from stress. Talaromyces pinophilus, on the other hand, produces plant hormones, auxins and gibberellins, which should promote plant growth, even under hydric stress. We will inoculate those fungi into young Quercus robur roots in vitro. We will check for colonization under a microscope, and then the production of metabolites. To check the effect of these fungi on the plant, we will also do a drought resistance test. To be simple, we will expose trees with or without these fungi to a drought stress. We just stop watering, and we will measure some parameters, like the chlorophyll content, to assess the well-being of each group. We hope to see that the inoculated trees react better to drought stress compared to the others. In conclusion, this project explores the role of endophytic fungi in enhancing oak trees' resistance to drought. The replication of these trees, providing an efficient solution for urban reforestation with more drought-resistant species, could contribute to sustainable urban greening.